The central dogma of molecular biology tells us that DNA makes RNA, makes protein. And so just to clarify, the information that's in DNA is transmitted or copied onto RNA in a process called transcription. And then the information that's in the RNA is used to make the corresponding polypeptide in a process translation. And so just to recap, each 3 nucleotides on the DNA are going to code for 1 amino acid. And so on the RNA, each 3 nucleotides as well code for 1 amino acid. And then in translation, those amino acids are put together to form a polypeptide, which then makes a protein. And right now we're going to focus on transcription. And so before we talk about transcription, we know a bit about DNA, but what is RNA? So RNA stands for ribonucleic acid. And it's very similar to DNA. So let's talk about some of the similarities. They're both nucleic acids. Both DNA and RNA are nucleic acids. And that means that they're made up of units called nucleotides. And each nucleotide has a sugar, a phosphate group, and a nitrogen base. So let's draw a nucleotide. So here's our backbone. So that would be the sugar and the phosphate group. That would be the backbone part. And then let's draw a nucleotide. I'm just going to draw a yellow nucleotide, which happens to represent thymine. So that would be our nitrogen base. So that's how DNA and RNA are similar. What about some of the differences between DNA and RNA? So I'm just going to make some room here. So by looking at it, the first thing that's obvious is DNA is double-stranded. And RNA is single-stranded, usually. There are some exceptions to this. There are some viruses that have double-stranded double-stranded RNA. But in general, RNA is single-stranded. Another difference between DNA and RNA is that the sugar in DNA is deoxyribose. And the sugar in RNA is ribose. And they're pretty similar, but let's just take a quick look at what deoxyribose and ribose look like. So the molecule on the left, right over here, this is deoxyribose. And the molecule on the right is ribose. And they're extremely similar to each other. The only difference between them is that in this spot right over here, deoxyribose has just a hydrogen, and ribose has an OH, an oxygen and a hydrogen. And so deoxyribose tells us that it's missing an oxygen. It's deoxy, without an oxygen that would normally be found in ribose. And so that's the only difference between deoxyribose and ribose. Let's go back to talking about the differences between DNA and RNA. So another difference between the two is that in DNA, one of the nitrogen bases is thymine, and it's one of the pyrimidines. And in RNA, wherever we would normally have a thymine, instead we have a uracil. And uracil is also very similar to thymine. It's also a pyrimidine, and we're going to take a quick look at these two molecules to see what they look like. So on the left, we have a thymine molecule. And on the right, we have a uracil. And the only difference between these two molecules is that in this spot over here, there's a methyl group and thymine, and in uracil, there's only a hydrogen. So you can see they're extremely similar to each other. There's one more difference I want to mention. DNA is more stable than RNA. And we want this because DNA is actually what stores genetic information in a permanent way. So we want it to be a very, very stable molecule that can hold on to the information for a long amount of time. RNA, on the other hand, its job is just to transfer the genetic information from the DNA to the ribosome to make our proteins. And so it's not as important for RNA to be as stable as DNA. And RNA is constantly made in the cell and degraded and recycled all the time, whereas DNA kind of just stays put. So now that we spoke about some of the differences between DNA and RNA, let's focus a little bit more on RNA. So here we have our RNA molecule. And then we're also going to just leave our DNA for now. And there are actually a number of different types of RNA. The first RNA we're going to talk about is mRNA. And that M stands for messenger. And messenger RNA is the RNA that's made during transcription. So if we were to transcribe this DNA, we would make a piece of mRNA. And the job of mRNA is to then go to the ribosome. And the mRNA is going to kind of run through the ribosome, something like that looks like this. And then the ribosome is going to put together the corresponding polypeptide chain. Hence the name messenger RNA. It takes the information that's stored in the DNA and then acts like a messenger by bringing that information to the ribosome so that the ribosome can put together the corresponding polypeptide. I want to just digress for a bit and talk about some of the differences in mRNA that you'd find between eukaryotic cells and prokaryotic cells. So in eukaryotic cells, mRNA is usually monocistronic. And that means that one piece of mRNA is generally going to code for one polypeptide chain. Prokaryotic mRNA can be polycistronic. That means that you can have one piece of mRNA that's coding for a few polypeptides. So you might have one part that codes for one polypeptide, and then maybe the next part codes for another one. Maybe it codes for a third polypeptide and a fourth polypeptide. And usually when you have a piece of polycistronic mRNA, the polypeptides that are being coded for are related in function in some way. Another difference I want to mention is where mRNA is made. So in eukaryotic cells, mRNA is made in the nucleus. And for that matter, transcription also happens in the nucleus. And I'm just going to abbreviate it like that. In prokaryotic cells, well, they don't have a nucleus, so mRNA is made in the cytoplasm. And, well, transcription also happens in the cytoplasm. So I'm just going to write also transcription. Just going to abbreviate it as well. And interestingly, in prokaryotic cells, translation can actually happen at the same time as transcription. So you can have a piece of mRNA being made, and while it's being made, it starts to attach to the ribosome and be translated. And the reason that that can happen is that they're happening in the same place. In eukaryotic cells, that can't happen. The mRNA is made in the nucleus, and then it has to leave the nucleus and go to the cytoplasm, where the ribosomes will then translate it. And the last difference I want to mention is that in prokaryotic cells, the primary transcript, I'm going to explain what that is in a moment. The primary transcript is mRNA. So the primary transcript refers to the RNA that's made during transcription. So in prokaryotic cells, the RNA that's made, that's mRNA. That's ready to go to the ribosome and be translated. Not so in eukaryotic cells. In eukaryotic cells, the primary transcript must be processed. And then after it's processed, well, it becomes mRNA. So only after it's processed is it messenger RNA and can it go to the ribosome and be translated. The next type of RNA I want to talk about is rRNA. And that R stands for ribosomal RNA. And ribosomal RNA is found in the ribosome. So the ribosome is made up of two components. It's made up of rRNA, and it's also made up of other polypeptides. And the interesting thing about ribosomal RNA is that even though it's a nucleic acid, it actually acts like an enzyme. And this is kind of odd because usually proteins act as enzymes and nucleic acids do not act as enzymes. So this is just an interesting fact to know about rRNA. And in fact, rRNA is known as a ribozyme. A ribozyme is an RNA that has enzymatic function. In fact, it's the enzymatic activity of the rRNA in the ribosome that puts together the polypeptide. The last type of RNA I want to talk to you about is tRNA. And that T stands for transfer. And transfer RNA has this 3D structure, and it looks a little bit like a clover. And the job of transfer RNA is to carry amino acids and then to bring these amino acids to the ribosome so that the ribosome can put together a polypeptide chain. Let's just recap. We spoke about messenger RNA, we spoke about ribosomal RNA, and we spoke about transfer RNA. And I just want to mention the enzymes that put together RNA in eukaryotic cells. So in eukaryotic cells, mRNA is put together by RNA polymerase II. Ribosomal RNA is put together by RNA polymerase I. And transfer RNA is put together by RNA polymerase III.